All right, thank you for joining us today. Today we are going to hear from Dr. Sergio Rutella on the immune correlates of TP53 mutational status in acute myeloid leukemia. So Dr. Rutella joins us from the Nottingham Trent University in the UK. He's a licensed hematologist and professor of cancer immunotherapy there. Over the last 25 years, Professor Rutella has been engaged in clinical and translational research focusing on immunotherapy approaches to improve the outcome of hematological malignancies, with an emphasis on antibody-based therapeutics and hematopoietic stem cell transplantation. His research programs focus on visualizing the state of cancer-immune interactions in individual patients and on biomarker discovery, with the aim to design rational immunotherapy approaches and to guide treatment choices. So thank you so much for joining us. We look forward to your presentation, Dr. Rutella.
Thank you very much for the kind introduction, and thank you for the invitation and for the opportunity to present today. So this is my disclosure slide. I have been receiving research support from Microgenics and NanoString Technologies, and this is relevant to my talk today. So my laboratory is currently working on the high-dimensional analysis of the AML immunological tumor microenvironment using targeted immune gene expression profiling on the Encounter platform, but also geomics, digital spatial profiling, DSP, to try and elucidate the topography of the immune infiltrate and how this correlates with immunotherapy responses. We also have an interest in identifying the oncogenic pathways that drive immune infiltration in AML, and as well as in solid tumors, including TP53 mutations, which will be the main focus of my presentation today. So just to provide you with a little bit of background about AML, so chemotherapy remains the standard of care for most patients with AML, despite recent approvals of novel drugs. Primary refractory or chemotherapy-resistant disease, so-called primary induction failure, PIF, as defined by not achieving complete remission, occurs in approximately 20% of the patient population. The 5-year overall survival from first relapse for patients with AML is only 10%. So the investigation of new molecularly targeted and immunomodulating agents remains a high priority for those in this space. And there's also an urgent need for predictive biomarkers to help identify patients who are more likely to respond to cancer immunotherapy. In this respect, the tumor inflammation signature, or TIS, has been shown to predict response to flotetuzumab immunotherapy in AML, and these results have been recently published by my group in Science Translational Medicine. So this slide summarizes our recent work that used immune gene expression profiling of 290 bone marrow samples from patients with chemotherapy-naive AML. This analysis identified 3 distinct gene expression profiles, namely an interferon dominant profile, including high expression of interferon gamma, PD-L1, PD-L2, inflammatory chemokines, and interferon downstream signaling molecules, an adaptive profile, including high expression of T cell, B cell, and natural killer cell markers, and the myeloid profile, including high abundance of polymorphonuclear cells, macrophages, and other cells of the myeloid lineage. These scores were then used in aggregate to stratify patients into an immune-infiltrated and an immune-depleted subgroup, as shown in panel A. Patients in the immune-infiltrated subgroup had higher levels of interferon-stimulated genes, T cell, and cytotoxicity markers, molecules implicated in antigen processing and presentation, and also actionable negative immune checkpoints such as PD-L1, BTLA, CTLA-4, and TIM3, as shown in panel B. The interferon-related RNA profiles improved our ability to predict chemotherapy resistance compared with the ELN cytogenetic risk classifier alone, as shown by the ROC values in panel C. We have recently used geomics, digital spatial profiling, to identify protein co-localization patterns with prognostic relevance in T-cell infiltrated AML. So this highly multiplexed technology allowed us to quantify 31 immuno-oncology related proteins in 10 bone marrow FFPEs from patients with newly diagnosed AML. So as shown in panel A, we analyzed 240 geometric regions of interest with varying degrees of T-cell infiltration, and we identified a prognostic 7-protein signature, which is referred to as signature 3 in panel B, and includes cytotoxicity molecules such as granzyme B, FOXP3, PD-L1, as well as STAT3 and BCL2. So when evaluated in silico, our protein signature was deregulated at the RNA level in approximately 25% of TCGA AML patients, who mostly clustered in the immune-infiltrated subgroup. And as shown in the mutation enrichment plot in panel B in this figure, the regulation of signature 3 genes significantly correlated with TP53 mutational status. And approximately 55% of the patients with TP53 abnormalities in TCGA AML had a missense mutation, as shown in panel C. AML patients with deregulated levels of signature 3 genes tended to have adverse cytogenetic risk, as summarized in panel A, and also had significantly shorter relapse-free survival and overall survival, as suggested by the Kaplan-Meier curves in panel B. So to summarize this first part of my talk, we identified an experimentally derived protein signature which is associated with immune-infiltrated AML with TP53 mutations and ultimately correlates with poor clinical outcomes. So is TP53 a new genetic driver of immune infiltration in AML? So let me provide you some background about TP53 mutations in this disease. So mutations of TP53 occur in 8 to 10% of de novo AML cases and are associated with chemotherapy resistance, with high risk of relapse, and with very poor prognosis, even after hematopoietic stem cell transplantation. The functional consequences of p53 mutation inactivation on host immunological responses have been largely overlooked in AML. And the p53 mutants studied thus far in AML do not show any evidence of gain of function mechanisms. So we asked the question whether p53 abnormalities correlate with the composition and functional orientation of the tumor immunological microenvironment in AML. To do so, we initially downloaded RNA sequencing data from TCGA, a collection of 147 cases of non-promyelocytic AML, including 14 patients with mutations of TP53. We then computed immune cell type specific and biological activity gene signatures as predefined linear combinations of relevant gene sets, which were then correlated with prognostic molecular lesions, including TP53 mutational status, NPM1 mutational status, FLT3-ITD, and clonal hematopoiesis of undetermined potential, CHIP, defining mutations in AXSL1, TET2, and DNMT3A, and with clinical outcomes. We also profiled primary bone marrow samples from 42 treatment-naive patients with TP53 mutated AML and 22 treatment-naive patients with TP53 wild-type AML using the pan-cancer IO 360 panel from NanoString Technologies. And finally, we analyzed primary bone marrow samples from 45 patients with relapsed refractory AML who were treated with flotetuzumab. 15 patients in this cohort had either TP53 mutations or 17p abnormalities with genomic loss of TP53. So panel A in this slide shows unsupervised hierarchical clustering of 44 immune gene signatures in 118 TCGA cases with non-promyelocytic AML and available information on prognostic molecular lesions, including 14 individuals with TP53 mutations. Two patient subgroups were clearly identified here, namely patients with high and low levels of immune infiltration, And compare with TCGA cases with favorable risk and intermediate risk molecular features, all patients with TP53 mutations showed high levels of immune infiltration and had higher tumor inflammation signature scores, inflammatory chemokine scores, interferon gamma signaling scores, and lymphoid scores, as summarized in panel B. And in particular, the expression of immune checkpoints PD-L1 and TIGIT and molecules associated with an immune-suppressed tumor microenvironment, such as the Treg transcription factor FOXP3, was significantly higher in TP53 mutated patients, as shown in panel C. And in line with previous findings, the Kaplan-Meier curves in panel D highlight that overall survival was significantly shorter, 4.5 months, in patients with TP53 mutated AML compared with 18.5 months in patients harboring other risk-defining molecular lesions. So we then profiled pre-treatment bone marrow samples, as I just mentioned, from 42 patients with TP53 mutated AML. As shown in the pie chart in panel A, most patients in this cohort had missense mutations of TP53, which are often associated with TP53 loss of function. Principal component analysis in panel B highlights that immune cell type specific scores and biological activity scores distinguished very clearly patients with TP53 mutated AML from individuals with TP53 wild type AML. As shown by unsupervised hierarchical clustering of immune gene signature scores in panel C, the frequency of TP53 mutated cases was 87%, 75%, and 8% only, respectively, in patients with high, intermediate, and low immune infiltration. And in line with our in silico analysis of TCGA data, immune checkpoints PD-L1 and LAG3, FOXP3, interferon gamma, CD8, and granzyme B, a molecule recently associated with features of exhaustion and senescence in AML, were more highly expressed in patients with TP53 mutated AML compared with TP53 wild type AML. So we next analyzed the immune transcriptomic profile at the gene level, and we identified a set of 34 differentially expressed immune genes between patients with P53 mutated and P53 wild type AML, as shown in panels A and B. And we will refer to this set of differentially expressed genes as TP53 immune gene classifier. The P53 immune signature genes have not been previously implicated in the TP53 pathway, as shown in Venn diagram, by the lack of any overlap with hallmark TP53 gene sets downloaded from the molecular signature database. So the differentially expressed molecules exhibited enrichment of KEGG pathways related to cytokine receptor interactions, autoimmune inflammatory processes, and IL-7 mediated and TNF mediated signaling, as shown in panels A and B. We then assessed the differentially expressed genes in silico, again for potential prognostic value in TCGA AML cases, and abnormalities of the 19 immune genes that were overexpressed in patients with TP53 mutated AML. And by default, this means RNA upregulation, gene amplification, deep deletion, and missense mutations, significantly correlated with worse relapse-free survival and overall survival estimates, as shown by the Kaplan-Meier curves in panel C. And by contrast, the 15 immune genes that were downregulated in patients with TP53 mutated AML carried no prognostic significance, and these data are not shown here. So next, we wanted to determine whether TP53 abnormalities identify a subgroup of patients with relapsed refractory AML that may benefit from immunotherapy with flotetuzumab, a CD123 by CD3 bispecific DART molecule for redirecting host T cells to AML in the CP-MGD 00601 clinical trial. So flotetuzumab is an investigational bispecific antibody-based molecule to CD3 epsilon and CD123 engineered in a DART format. And flotetuzumab binds both tumor and effector cells to promote the formation of an immunological synapse and to redirect polyclonal effector T cells to kill AML blasts in a major histocompatibility complex independent manner. So this slide summarizes the clinical characteristics of the 45 patients in the flotetuzumab cohort. Median age at time of study entry was 61 years. Approximately 75% of the patients had an unfavorable cytogenetic risk based on the 2017 ELN classification. And 15 patients in this cohort had either TP53 mutations or deletions of 17p, to which P53 is mapped, that are usually associated with loss of one allele of TP53 and mutation loss of the other. And response in this study was assessed by modified IWG criteria, and specifically complete response, less than 5% bone marrow blasts, were defined as either complete remission with partial hematopoietic recovery, complete remission with incomplete hematopoietic recovery, or morphologic leukemia-free state, MLFS. No response was defined as either treatment failure, stable disease, or progressive disease. So baseline bone marrow samples for immune gene expression profiling were available in 13 out of 15 patients with TP53 mutations and or genomic loss of TP53. Among these, 10 patients showed intermediate to high immune infiltration, as highlighted by the heat map in panel A. Time on treatment and time to patient death and or censoring are summarized in panel B for patients with TP53 mutations and or 17p abnormalities, including 3 patients who proceeded to receive allogeneic hematopoietic stem cell transplantation. So the overall response rate was 60%, 9 out of 15 patients, with 47% of them achieving complete response, as defined by less than 5% bone marrow blasts on study. And in responders with P53 mutations, median overall survival was 10.3 months, with a range from 3.3 to 21.3 months, suggesting that flotetuzumab may alleviate the negative prognostic impact of TP53 abnormalities in AML. So the reduction of bone marrow blasts in 14 patients with TP53 abnormalities for whom a post-cycle 1 bone marrow sample was available averaged 51.2%, as shown in panel A. In patients with mutations of TP53 and with evidence of response, the tumor inflammation signature, interferon gamma, Treg, CD8, and exhaustion gene expression scores were significantly higher at baseline compared with non-responders, as shown in panel B, highlighting the association between response to T-cell engagers, such as flotetuzumab, and the T-cell inflamed tumor microenvironment. We also identified a set of genes that were differentially expressed between patients with TP53 abnormalities and those with P53 wild-type AML, and that were associated with response to flotetuzumab. Interestingly, CD8b was the top-ranking gene associated with response, with an ROC value of 0.879, as shown in panel B. And finally, flow cytometry experiments performed by Mike Reddick at University of Washington using bone marrow samples from patients on study showed a trend toward higher percentages of CD8 T cells in individuals with mutations of TP53, therefore validating the transcriptomic findings of this study. So just to conclude my talk, immune transcriptomic analysis of in silico and wet lab cohorts of TP53 mutated AML suggests the presence of high T-cell infiltration and high expression of immune checkpoints and interferon gamma signaling molecules compare with AML subgroups with other risk-defining molecular lesions. Experimentally derived TP53-related immune gene signatures are significant for survival prediction in AML. Our translational study has shown that immunotherapy with flotetuzumab has efficacy in individuals with altered TP53 status, with an overall reduction of bone marrow blasts averaging 15.2% and 51.2%, and an overall response rate of 60%. We believe that these findings encourage further studies of flotetuzumab in larger cohorts of patients who harbor this molecular lesion. So this is my acknowledgement slide. I wish to express my thanks to co-authors, collaborators from many institutions around the world, and also funding sources. I will be very happy to take questions if you do not mind emailing me. Thank you very much for listening.
That was fantastic. Actually, I have some questions as well for you, Dr. Rutella, if you can answer. Perfect. So what is the recommended input of RNA for the analysis of bone marrow samples?
Yeah, so we generally used approximately 150 nanograms per ml from unmanipulated bone marrow samples. And our samples were always looking very, very good in terms of QC. So we almost never had any QC issue with this amount of RNA.
Perfect. And did you use purified leukemia blasts in your experiments?
No, we didn't. So we used unmanipulated bone marrow samples because the aim of this investigation was to gain insights into the composition of the tumor microenvironment. So we didn't want to work with purified AML blasts to do this.
Great. Is there any evidence from solid tumors that TP53 mutations associate with response to immunotherapy?
Actually, there is evidence in lung cancer that patients with mutations of TP53 may respond to immune checkpoint blockade. Yes.
So, Dr. Rutella, this is pretty exciting times with the whole spatial, revolution. You can do bulk gene expression analysis, adding on spatial findings. I'm curious to hear your thoughts on these techniques and, how they're related and integrated, and how you would apply these types of technologies moving forward.
Yeah, so this is a great question, and it's a very interesting point. So we are trying to integrate molecular lesions which are established prognosticators in the clinic with the immune subtype. We have generated a considerable amount of information suggesting that immune subtypes may predict resistance to chemotherapy and also response to immunotherapy. So I think it will be very important to validate these findings prospectively. And of course, using the NanoString approach has several potential advantages, including the rapid turnover for this 24 to 48 hours, and also the ability to customize your own panels. You may want to include, as I just mentioned, established molecular prognostic lesions, as well as genes that capture the functional orientation of the immunological tumor microenvironment. So I really look forward to the translational elements of this research.
Fantastic. And then finally, are there other genetic drivers of cancer associated with a hot tumor microenvironment?
So yes, in our study, we have identified RANKS1 mutations as being associated with the hot tumor microenvironment. And again, RANKS1 mutations correlate with poor clinical outcomes in AML. And there is evidence published by others in solid tumors, and specifically in lung cancer, that KRAS and NRAS mutations may be associated with a hot tumor microenvironment.
Great. Thank you so much. I really enjoyed your talk and really appreciate you sharing your research with us. This was fantastic.
Thank you very much.